Okay, we're on to the final part of this section. These are some great pictures. It's a good summary. It's showing transcription factors. So here is a double-stranded section of DNA, and it's showing a gene right there. RNA polymerase is an enzyme involved with the copying of DNA. In other words, making a strand of messenger RNA. See right here, well, you can see it's being split. And then see this coming off. It says messenger RNA transcript. And it's in the form of codons, the 3 sections of nucleic acids. Or, well, nitrogenous base, I should say. Here is the DNA section. See the T, the G, the C, and then right here there's an opposite complementary strand of messenger RNA being made. So opposite the A, there's a U. See, this is placing an A across from the T. All this is accomplished by enzymes. And it's going to peel off like that, and then it's going to get to the end of a gene, and that's where it will stop. All right, showed you that. Okay, now this actually happens inside the nucleus of a cell. This whole transcription portion, that happens in the nucleus. When it moves out into the cell cytoplasm and attaches to a ribosome, and it's going to start being translated at that point. So it says large ribosomal subunit. You can see where they're kind of separated. It doesn't actually pick up like this. It's just showing here is a transfer RNA molecule. See, there's the MET start codon. And it says initiator transfer RNA is going to start the process of translation. You're translating the genetic code. And then here, like there's MET, here's glycine. See how it's coming along and it's dropping onto here. And then what happens, these transfer RNA molecules, they will break off and go back and go get other amino acids, depending on what the genetic code is. So it's going to read, see here's AUG, which is the start codon, GGC, and then it's going to read, that's GGC, this is GCC. It will keep reading the genetic code and then going out and getting the appropriate amino acid. Okay, just like that. Just a little bit bigger picture. See, this is showing, see here, this one's peeling off. So methionine, glycine, alanine, it looks like. And then it just keeps going on as long as the genetic code continues until it comes to a stop codon is when it will officially stop. But there's one that looks like proline, phenylalanine, there's tryptophan. And then here it says a stop codon, UAG. And let's see if that's actually right. There it is, UAG, stop codons. At that point, the whole process will stop. Now, here is a summary, and then we'll get to a little bit of practice, and that'll be the end of this. So right here it says transcription. Like I said, this all takes place. Remember, this is the nucleus of a cell. There's the nuclear membrane or envelope. Here's the rough endoplasmic reticulum. And so basically, here is the strand of, it says messenger RNA transcript. Now, don't worry about introns and exons, Those are sections that get removed. But that'll be for another day. So here's your messenger RNA strand right there. You see it's moving out. Remember, this membrane is selectively permeable, allows certain things to cross, and things can't. And so once it moves out here, it's going to attach to a ribosome, and then here comes the transfer RNA, and of course it's going to, the first thing it's going to grab is the MET start codon, and then it will start building a polypeptide. And like it says over here, it binds to the ribosome and is translated by transfer RNAs, producing a polypeptide. There's the polypeptide, and then, like it says here, a modified functional protein. And that's the whole process. So you start in the nucleus, it moves out to a ribosome, and now you have a protein. If you're wondering what are proteins, those are enzymes. Sucrase, maltase, lactase. enzymes that help break down food, enzymes that power metabolism, all that stuff we covered with energy conversion, the cytochrome C oxidate, all that stuff that's part of the electron transport chain, Krebs cycle, alpha ketoglutarate, all those things are enzymes. Okay, so now for some practice. You are going to do this in your lab. If I can get I'm going to make this a little smaller, get the line up. There we go. So here is a strand of DNA, and then we're going to make an opposing strand of DNA, and then we're going to transcribe it here, and then depending on what that code is, we will translate it here. And we'll do that next. Now, here's opportunity to practice, to put everything together, kind of join all the information we just went over together, and we're going to now we're going to do transcription first and then translation. First thing we're going to do, so DNA, copy DNA. So we're going to make an opposing strand of DNA. And what you would do, here's a section of DNA. This could be a gene. Genes are, I think the shortest human gene is about 1,000 of these, but this one's pretty short. So first thing, you're going to fill in what is called the opposite corresponding nitrogenous base. So ATGC, adenine, thymine, guanine, cytosine. So you want to fill in on the opposite side the corresponding base pairing. A's always bind with T's, and T's bind with A's, vice versa. G's always bind with C's, and then C's always bind with G's. So what you would do, so I'm going to put a T right there opposite the A, so adenine binds to thymine. Then put another A. Opposite the G, guanine. Of course, that's going to bind to cytosine. Opposite the C, you're going to put a G, if you see how this is going. So one more, and then adenine. Of course, you're going to put a T binding to thymine, and then fill out the entire DNA strand. Okay, we have now filled out the entire strand, and if you count each base pair, there should be 24 on the top, 24 on the bottom, and we've filled in the corresponding DNA. You match it with the corresponding nitrogenous base. So next, we're going to do transcription. So what that means is this section of DNA, we now want to make a strip of messenger RNA. Remember, RNA has 3 base pairs that are the same, but instead of thymine, RNA, it gets replaced in RNA with uracil. The reason that is a cell synthesizes RNA, and thymine costs more metabolically to make than uracil, and uracil still accomplishes the same thing. So think of uracil being like the off-brand. So what we would do, put an A opposite the T, and now opposite this A, where you would normally in DNA put a thymine, you're going to put a uracil. Opposite the C would be a G, and then you fill in the next few. G would be C, and then opposite the T, you're going to put an A. Then opposite the A, you're going to put another U. And then fill in the corresponding messenger RNA. Well, you fill in the corresponding bases, but you're going to make a messenger RNA strand, just like it says right here. All right, now we have finished filling in our messenger RNA strand. If you count each base pair, in our particular example, it should be 24. Now, these represent the transfer RNA molecules that are going to move out into the cell cytoplasm and attach to the correct amino acid. And so what happens is this messenger RNA strand is going to go out and attach to a ribosome, and the ribosome is going to start translating the genetic code, which these are in 3s, they're called codons. And these transfer RNAs would then go out and attach to depending on what this code says, the codon, the triplet, the correct amino acid. So that's where this chart comes in. Now, starting off, so you have a start codon, this MET, methionine, or methionine, I've heard it twice, both ways. See the AUG? Okay, that transfer RNA is going to go out and attach to that. So you can put here MET, could have put start, CAU, going back to our chart, too much caffeine, CAU, there it is, histidine, HIS, so it will bring it back to the site of the ribosome, and it's going to drop the amino acid in the sequence. These are transfer RNAs. So we're gradually building what is called a polypeptide. We're making a protein. The next three, CUG, CUG, leucine, CUG, yeah, LEU, wrong button. There we go. What are the next three? And you would do this for the entire thing. So you would then go in and fill out the corresponding, put the correct amino acid depending on what the codon is. ACU. ACU. THR. Okay, ACU, CCU, proline, UUC, phenylalanine, CAG, glutamine, this, oh, MET. In 3, 2, 1. All right, I have now finished filling in the corresponding, well, the amino acids. These were transfer RNAs, and they went out and grabbed all of these amino acids, and that's translation. This was the start codon, and see this UAA? There are 3 of these. UAA, UAG, and UGA. They represent what are called stop codons. That's how the ribosome knows to stop reading the messenger RNA strand. And now you have a fully functioning protein. Ours is really not that big, of course, but you think about a protein like hemoglobin. Hemoglobin is 256 amino acids. So we did 1, 2, 3, 4, 5, 6, 7, 8. So ours is pretty small. I went ahead and spaced these out so they would match up with the transfer RNAs, and I did the lowercase just like in our chart. But that is transcription and translation in a nutshell. It's really interesting, and in your lab you'll be doing this. Okay, I hope that helped. Just kind of won't go through, and it's a little hard to do on a computer. It's better to write it out, but oh well, got it to work. The last thing to add here, gene regulation refers to the mechanisms that act to induce or repress expression of a gene. So in other words, all of this is controlled. And when there's a loss of control is when you have, things such as cancer or mutations or, certain developmental disorders and things like that. I'm thinking of trying to give you an example. Muscular dystrophy, which a person with muscular dystrophy, their skeletal muscle, when they contract, do not stay uniformed and instead sort of go in all directions. So it results in inability to move their arms and legs and skeletal muscle in a coordinated way. Well, it makes a person look like they're kind of all bound up. Well, they're missing a protein called dystrophin, which stabilizes the sarcolemma when a muscle contracts, the cell membrane of a muscle fiber, skeletal muscle fiber. So just missing one little protein, it's a genetic disorder, causes all that trouble. So yeah, it is highly regulated. Okay, I hope these videos help. You're going to be doing, I have one for lab, and I have just an overview one for you. So anyway, thanks for joining me.